Welcome, students, to our live class. I'm your instructor, Tahmid. I'll be teaching the A-level segment of biology, both Cambridge and Edexcel. I try my best to handle this live session, trying to be as clear as possible and trying to make difficult concepts easy for you. So let's get started. So, I would like to talk about a little bit about how your mindset and how your approach is supposed to be when you're studying biology. You see, biology it has concepts. As well as concepts, you have to have a very good understanding of the things you need to memorize, the things you need to identify, and most importantly, how you write things in the exam. These things are very important, like how examiner writes the question, understanding the question, reading the question carefully, and answering the question according to its merit. So I'll just go over to some of the tips that I've noted down on the slides over here. So let's go through them, shall we? So first of all, understand before memorizing. Memorization can only take you that far. But if you understand it, you understand what points to mention, specifically what points to mention in the definition, or let's say in the question, that's gonna go a very, very long way. And that's gonna be the secret for you gaining marks in the exam. So, for example, don't just memorize that enzymes are affected by temperature. Understand why high temperature denatures the enzyme. By breaking hydrogen bonds, or let's say changes in pH, changes the active site, or you can use and in case of temperature, denaturation happens. So you see this word denaturation, active site changes. These little points, you have to understand them in order to answer them properly. Print the syllabus, tick off learning outcomes as you revise. A very underrated it's an underrated tip. The syllabus, you can use it as a checklist, and the syllabus enforces you to make sure you haven't missed anything, especially in AS. AS syllabus is very vast, so it's pretty important you have an idea of almost each and every content in the syllabus, because questions can literally come from anywhere. All right. If the syllabus says describe and explain, you must be able to do both. Remember, describe and explain is two different things. Describe, for example, describe the structure of plasma membrane. So when they say describe, you essentially will start listing the features of cell membrane. For example, phospholipid bilayer, it is hydrophobic, it has carrier proteins. Point out the points in a bullet form, in a bullet system, like how you list features. And here, look here, explain its function. And when they ask explain, you have to link the structure to the function. Explanation is not more about you listing points. It's more about you write elaborately about that question. Along with, you have to correlate. For example, this bacteria has cell wall. Cell wall is made up of peptidoglycan. Why is it made up of peptidoglycan? Because it provides support, structure, and protects the bacteria from different outside trauma or inside metabolic issues. So link the structure with the function. Also, the examiners are very strict with command words. So if the question comes around state, so you have to write short answers with no explanation. These ideally come for 1 to 2 marks. And when they ask you to describe it, you have to, first of all, explain what you see, ideally in graphs. When you're explaining graphs, you have to see the question, you have to see the points, which from point A to point B it increases, from point B to point C it decreases. You have to mention from point A to point B, the graph increases due to dash, and then B to C it decreases due to dash. All right, students? So Just describe. So when the question says describe, just explain what you see, and no need that further reasoning. Or when the question says explain, then that's where you have to give reasoning, and reasoning comes from memorization, having a good idea of the concept and the points that you need to mention. That happens by understanding and practicing past papers. Use biological principles, so give reasons using biological principles. And when the question comes about compare, similarities, differences, examples. Okay, students, let's go next. Learn definitions, word for word. This is very important for biology. Definitions are free marks. All right. I always say this to all my students that definitions are supposed to be free marks because you will memorize it and you jot it down in the exam day, and you get free 2 to 3 marks or 1 mark. So definitions are key. So, and the way you write your definitions is also very important. For example, osmosis. Osmosis is water moving through a membrane. You will not get a mark, unfortunately. But if you say osmosis is the net movement of water molecules from a region of high water potential to a region of lower water potential through a partially permeable membrane. See how beautiful the definition is. You have mentioned the concentration difference, high to low. You have also mentioned the partially permeable membrane. So these points are what's gonna give you the marks. Use correct biological terms. Very, very important. Understand your biological terms as well as apply it when necessary and in correct context. Never say don't say slangs stuff or like short forms like BCZ instead of because. Don't do that. All right. Make it as easier for the examiner to check your question paper, and thus increasing your chances of getting marks. So don't say the words such as things, stuff, moves across. No, start use the words diffuses, is transported, passes through via proton channels. So you guys have to up the game here. Link the structure to the function. chromosomes. So what happens in mitosis, as I said earlier, the nucleus is replicated and duplicated as well as the chromosome. And cytokinesis involves the cytoplasm and the organelles between the two cells. The number of chromosomes doubles and the number of organelles doubles, so that the single mother cell can divide into two separate daughter cells. Most body cells are diploid. Before mitosis, DNA doubles to 4n and then divides. Each daughter cell returns to 2n. Now, what does this exactly mean? Let me just draw it out for you guys. So what happens is, imagine this is the cell and this is one chromatid, alright? This is one chromatid. Now, a chromosome has two chromatids. So basically a chromosome has two pairs of chromatids. Okay, so this is two pairs, and then we got another two pairs. Now, what happens is, to form these two chromosomes, we needed how many chromatids? We needed 4 chromatids. So 1, 2, 3, 4. So from 4 chromatids, we got 2 chromosomes. So basically, this is 2n and this is 2n. So 2n plus 2n is 4n. So what happens, once we form the 4n, which is the important part, the cell divides into 2 cells. Now each cell, so basically this gets separated, so we'll have 2n on one cell and then 2n on the other. So let me simplify it again. Each chromosome has 4 chromatids, and in this case you have 2, 3, 4. You have 1, 2, 3, 4, 5, 6, 7, 8. 8 chromatids that has been formed due to these 4 has been formed extra due to the replication, due to mitosis or DNA replication. This has been formed due to mitosis from these 4. Then these 4 eventually causes the formation of the extra chromosome, and then this one eventually this 2n plus the previous 2n forms the 4n, and then the 4n gets separated into 2 different cells. After mitosis, what happens? 2 cells are formed. Then this 4n, from this 4n, 2 stays in one cell, and then the rest 2 goes to the other. So in basic terms, this is the initial chromosome and this is the second one. So I have just turned this into red to make it easier for you guys. I'll turn this into red as well. Alright, hope it makes sense. Alright, I will suggest you guys take a screenshot or take your time with this diagram. Okay, students, just remember the ones that are in red are produced due to mitosis. If it's in red, produced due to mitosis. Alright, hope it's much more clear now. Alright, so I'm just going to reread this line since I've given the explanation. Most body cells are diploid. Okay, 2n. What happens after mitosis? It becomes 4n. And then what happens when the 2 cells are formed? 2 cells both will have 2n and 2n. Basically, it returns to 2n. Now, let's learn about the stages of mitosis. Mitosis can be divided into 4 stages. You got the prophase, you got the metaphase, you got the anaphase, and you got the telophase. 4 different stages, 4 different things happen, completely different things happen in each stage. So I'm just gonna read the theory for now, and then we're gonna go to the diagram. So what happens in prophase? I want you guys to pay attention in here. The chromosomes condense. Chromosomes condense. Nuclear membrane, that is the membrane of the nucleus, breaks. And then what happens? We have something forming, which is called the spindle or spindle fiber. Some of you guys might be familiar with it as spindle fiber. Now, what does this? So what happens in prophase? Chromosome condenses. The nuclear membrane of the nucleus breaks. Spindle fibers form. What happens in metaphase? All the chromosomes that has condensed, remember, it appears in the equator, basically center of the cell. Chromosomes line up at the cell's equator. Now that the chromosomes are now at the center of the cell, what happens? It has to go to the poles, and we call this phase anaphase. What happens? The sister chromatids are separated and moved to the opposite poles. So when all the chromosomes are concentrated in the center of the cell, in the anaphase stage, the sister chromatids go to the opposite poles, basically right and left, or north or south, whatever makes it easy for you. And then in telophase, what happens? The nuclear membrane finally gets that was broken, it reforms, and then the chromosomes decondense, and then the cytokinesis follows, basically production of cytoplasm and the organelles, and essentially the two cells become separate. Let me show you a diagram. Okay, interphase is where we are preparing the whole cell for the M phase or mitosis phase. So you have the prophase. As you can see in the prophase, let me take the pen out. As you can see in the prophase, you see these fibers? Yeah, these are spindle fibers. These are spindle fibers. And I want you to notice something. There is no nuclear membrane. So the nuclear membrane has broken. So nuclear membrane broke. And what happens to the chromosomes? They condense. This was the prophase. Now let's come into the metaphase. All the chromosomes are in the center. All the chromosomes, what they do? They undergo, they all come into the center. As you can see, they're all in the center. In prophase, the chromosomes condense, and in metaphase, all the chromosomes come at the equator. So I like to say it as chromosomes come towards the equator. Now let's learn about the anaphase. In the anaphase, what will we learn? Sister chromatids go towards the opposite poles. In this case, we can see the chromosomes are going to the opposite poles. And another thing you can notice that you can see this pinching. You can see this pinching is happening. There is a depression. There is a depression here. Eventually, what happens is, I will try to demonstrate. Imagine this is the cell. So what happens? A slight pinching happens, right? And then eventually, the pinching becomes greater and greater, the depression becomes greater, and then the two cells separate. And then we'll learn it in telophase. For example, look at the telophase. See, the depression is greater, right? And then what happens at the end of the telophase? You have two separate daughter cells. So what happens in the anaphase? Sister chromatids move towards the opposite poles. Imagine it as the great stretch. All right. Now let's learn about the telophase. In the telophase, cytokinesis happens. All right. Basically, I'm just gonna write it here. Okay. What happens in telophase? Cytokinesis. Cytokinesis happens. Why cytokinesis happens? Basically, cytoplasm in numbers. So there is a replication and duplication of cytoplasm and the organelles, because obviously we need it for the two daughter cells that are going to be produced. And then what happens? We can see the nuclear membrane forming, slightly nuclear membrane is forming. So the nuclear membrane. Alright. Oh. Nuclear membrane is forming. And then another thing I can see is the membrane and the chromosomes. They are decondensing. So as you can see, the chromosomes are decondensing. Where can I write it? I'm just gonna write it here. So the chromosomes are decondensing. Okay, and then we have two beautiful daughter cells. I hope this whole process clicks for you guys. So let's slightly recap. Interphase is where it prepares for the M phase. M phase has how many phases? 1, 2, 3, 4. 4 phases. Mitosis phase. Basically, M phase has basically mitosis and cytokinesis, but mitosis has 4 phases. So, prophase, anaphase, prophase, metaphase, anaphase, and telophase. 4 phases for mitosis. And M phase has how many phases? 2. Mitosis and cytokinesis. So what happens in prophase? Spindle fibers are formed, the nuclear membrane breaks, and the chromosomes condense. Metaphase, they all come into the equator. Chromosome comes towards the equator. Anaphase, sister chromatids stretch out into the opposite poles. And telophase, all these things happen. Cytokinesis happens, nuclear membrane forms, and chromosomes are decondensing. And then daughter cells are formed. Students, 1 minute. I'm coming back in 1 minute. Alright, students, I'm back. Let's continue. Alright, we learned a lot about mitosis, didn't we? Let's learn about why we need mitosis. What's the importance of mitosis? So I condensed the whole Markey scheme points into a single sentence. So mitosis is essential for growth. 1 point, repair, 2nd point, and cell replacement. 3rd point. So it's important for growth, repair, and replacement. Of what? Cells and tissues. Of cells and tissues. Okay, students, Now let's read a little bit about meiosis. Meiosis is slightly less important topic. The questions usually come in the form of MCQs. You just have to know slightly a little bit about the features, and I'll show you I'll show you the diagram just to make the things a bit more simpler. But the diagram isn't necessary. You just need to know the features and ideally the definition. All right, students. Okay. Alright, meiosis. What is meiosis? It's a specialized cell division that produces four haploid gametes. Haploid. What's the word haploid? In mitosis, we used to produce what? Diploid. And in meiosis, we produce what? Haploid gametes with half the original original chromosome number, and it has two divisions. So meiosis is nothing but production of four haploid gametes with half the original chromosome. So if the original chromosome was, let's say, 47, half that will be the haploid. And if, let's say, the initial is 22 chromosome, half that is going to be 11. Let's say 11 is going to be the haploid chromosome. Now, what are the types of meiosis? We have type 1, and we have meiosis 1 and meiosis 2. What happens in meiosis 1? The homologous chromosomes pair, exchange the genetic material, and separate. So the homologous chromosomes, they pair together, they exchange the genetic information, and they separate. So basically two chromosomes, alright, homologous, basically they're of same type, they pair together, they exchange their information, and then they separate. That's meiosis 1. Meiosis 2 is similar to mitosis, where the separating sister chromatids will replicate. Alright? We'll learn this in the next picture. Now, why do we need meiosis? Why are we learning meiosis? It's very important for sexual reproduction and genetic diversity. Now, how does meiosis contribute to the genetic diversity? It's very important. I'm just going to write it down. My meiosis. Okay, I'm just writing it down here. Meiosis causes genetic diversity by crossing over so it causes genetic diversity by crossing over and sharing of alleles. So it ensures the chromosome numbers remain constant and remains the same and constant across the generations. So it's very important where this question usually comes in the exam, how meiosis causes genetic diversity. So whenever it comes, you just write this down. Meiosis causes genetic diversity by crossing over and sharing the alleles. All right. I think so this is the picture of the meiosis. Let's come in. So interface is, you guys are familiar. Interface is where we prepare the cell for what's about to come. So either meiosis or mitosis. The interface is where we prepare the cell. Meiosis 1 is where the chromosomes pair up, exchanges the genetic material, and then they separate. So you have these chromosomes and then they exchange. As you can see, the result of exchange, we have a mixture of black, and then you have a little bit within the black you have a little bit of white, and within the white you have a little bit of black. And as you can see here, within the white you have a little bit of black, within the black you have a little bit of white. So as you can see, there is a sharing of the genes or alleles that has occurred. And then what happens in meiosis 2? You have replication. So four daughter cells with haploid number of chromosomes. Why haploid? As you can see, there is one chromosome has how many chromatids? 4, right? How many chromatids are in here? 2. That's why we call it haploid. So this is one haploid, another haploid, another haploid, another haploid. Right, next move on. Well, remember telomeres that was present at the end of the chromosome? Like you had a chromosome at the end of the chromosome, there was telomeres. Now, why do we need this telomere? So I'll tell you guys some facts from research. So we have found that the number one reason why we age and why we die from aging is because of these telomeres. And research has said that the secret to long life could actually be preventing the shrinkage of the telomeres. What basically these telomeres, when a cell replicates, when a chromosome replicates, between these replication, each replication, slight amount of telomeres or essential genes gets left out. What happens eventually that these telomeres become shorter and shorter and shorter as you age. So as you get shorter, the telomeres get shorter, so does your life expectancy, and that's when you start aging. So ideally, if you think about it, if we can stop the shrinkage of the telomeres, that could be the secret to living long, right? I guess we have to let science, figure it out. All right, let's get back to our actual content. So the telomeres act as a buffer region of non-essential DNA. Basically, up to telomere, all the DNA is essential. After telomere, everything is non-essential. They ensure that no important coding sections near the ends of the DNA molecules are left out. Okay, so telomeres make sure all the essential genes that are present in the chromosomes are replicated. But unfortunately, as I told you, over time, the telomere shortens, and eventually we keep losing those essential genes. This telomeres ensures no genes are lost during cell division. So, what's the function of telomeres? To make sure no genes are lost and to make sure all the essential genes are replicated. But unfortunately, telomere stops working properly. Alright, let's move on. How tumor occurs? Tumor is a swelling or lump of mass in our body. Now it happens mainly, the main cause of tumor formation is the mutation. Now, what are the causes of mutation? Just read it out. Errors during DNA replication, as I said, remember, telomeres. Spontaneous chemical changes in DNA. Let's say you get exposed to different chemical hazards where the DNA sequence gets disrupted. Failure of DNA repair mechanism. When our body system doesn't does the cell tissue repair properly. Transposable elements, example, jumping genes. Let's say, for example, genes are just not acting right. They're moving sporadically in different directions. UV radiations, very important. UV radiation, X-ray radiation, all types of radiations. Radiations are not good. They are the number one single cause of mutations. Good example could be Chernobyl. When the nuclear plant exploded, the radiation started causing people cancer, or the Hiroshima Nagasaki incident when the nuclear bomb fell caused cancer to many people. It can even happen to workers who work in the radiation sector, work with nuclear, let's say some nuclear elements, who work in X-rays, etc. So X-rays and gamma rays, and then chemical mutagens like toxins, pollutants, and drugs. Now you might be wondering, are all tumors bad, or are all tumors cancers? No, the tumors are of two types. Can be characterized into two types. Benign and malignant. And malignant. Benign is a tumor, malignant is also a tumor. The thing about benign is it's non-cancerous. Alright? It's non-cancerous and does not spread. And doesn't spread. But malignant tumors are cancerous and they do spread. Okay, students? Now let's learn a little bit about malignant tumors. So malignant tumors, how are they cancerous? They interfere with the normal functions of the organ or tissue. They in which they have started to grow. Example, they may block intestines, lungs, blood vessels. Basically, they interfere with organ function and tissue function. The malignant cells can break off the tumor and travel through the blood or lymphatic system to form secretory growth in other parts of the body. So it can metastasize, it can grow and move into different parts of the body and thus start spreading there. So, very important. Malignant tumors. Alright, let's solve some questions. Alright, let's start. The diagram below shows the nucleus of a cell during mitosis. You have a cell during mitosis. Now, you have to identify the structures A, B, and C. I think the question just turned a little bit weird, but essentially this is supposed to be A, and this is supposed to be C, and this whole thing is B. Now, what is A, what is B, and what is C? A is pretty much understandable. It's a pole. It's the pole. And B, the whole thing, is a chromosome. How did I know it's the chromosome? Because as you can see, these things, these are the chromosomes. So my bad, I think it's supposed to, I should just put this here. There you go. This is B, alright? So this will be chromosome. This whole thing is a cell, alright guys? And then what is this? This is the spinal fiber. So C is spinal fiber. Okay, students, so this whole thing is a cell. This is the chromosomes. These are the poles, and these are the spinal fibers. Okay, let's erase. Alright, name the stage in which each of the following processes take place. So where does the condensation of the chromosome happens? Anaphase. Oh, sorry, prophase. The condensation of the chromosome, where the chromosome condenses, is the prophase. Movement of daughter cells to the poles, the stretching, anaphase. Alright. Let's go to the next question. Now, the substance can be used specifically to inhibit the formation of spinal fibers. So stops mitosis at a particular stage. At which stage would colchicine stop mitosis? So it is a substance that stops the formation of spinal fiber. So if spinal fibers are not formed, then the chromosomes cannot go towards the pole. So at which stage would this stop mitosis? It's gonna be the metaphase. Because if this happens, then the metaphase cannot happen. If there is no formation of spinal fibers, then the chromosomes cannot come into the equator. And if the chromosomes don't come into the equator, then the chromosomes cannot go to the poles. So essentially, metastasis will not happen first, and then anaphase will not happen. But metastasis comes first. So metaphase will be the answer. Name two precise locations where mitosis occurs in plants. So yeah, you can think of the root tip, or you can think of the root tip. You can think of the just overall plant cell for plant cell repair. Right. Let's move on. All right, outline the sequences of changes that are undergone by chromosomes during mitosis. So first of all, we have to mention the chromosomes replicate. So chromosome replicate into chromatid. And then DNA deposits on chromosomes, and then the chromosomes condense. So essentially the chromosome condenses. And then it gets attached to the spinal fiber, and then what happens? The complete replication happens. Binds to spinal fiber and then comes to the equator. What happens when it comes to the equator? The chromosomes move to the poles. So the chromosome moves to the poles. And then essentially, I think it's pretty much it. Yeah, the chromosomes replicate into chromatids, they condense, and then they bind to the spinal fiber, goes to the equator, and then, we can also say in telophase. What happens in telophase? The chromosomes duplicate. So we can also say move to the poles, and then chromosomes. We can say chromosomes duplicate as well. Yeah. Oh no! Chromosomes it condenses, right? So decondense. Yeah. In telophase, the chromosomes decondense. Okay, that was a big question. Well, this diagram. Let's look at this diagram. It's pretty obvious. This is telophase. Briefly describe the events of the next stage of cell division. Cell wall forms. Or you can say the vesicles. Vesicles from Golgi complex can form. Spinal fiber disintegrates. So 2 marks. So I will start with spinal fiber disintegrates. And then I can say the cell wall forms. because this is a plant cell, remember? Alright, next question. Right, what happens in prophase? Chromosomes become shorter and thicker. So I will instead of writing the whole thing, I'm just gonna write number. Okay, so this is 1, this is 2, this is 3, this is 4, this is 5, and this is 6. So chromosomes become shorter and thicker. That's gonna be 1. And then what happens in metaphase? So it attaches at the equator. So this will be 2. So what happens in anaphase? Anaphase moves to the pole. So it's gonna be 3. And then what happens in telophase? Nuclear membrane reappears. So 4 chromosome. And then what happens in interphase? Chromosomes will replicate except at centromere. So that's the most accurate answer I can think of. And obviously cytokinesis is the replication of cytoplasm. So 6. Alright, let's move on. Okay, here's another question. Yeast cells reproduce asexually by a process called budding. Budding is the replication process of the yeast. The parent yeast cell produces a bud. Explain the importance of mitosis in budding. So it's essentially it's going to be around 3 marks. So first of all, mitosis is used for the growth of cell. Cell growth happens. And then we can say you can say same genetic material. There's no differences in genetic material, so we have the same. All the daughter cells will have the same genetic material. So I can say no variation. Okay, we can say no variation. Also, we can say it's easier for, let's say, it's more efficient for bread making. Yeast is used in bread making, right? So we can also use it's efficient for bread making. Alright guys, alright, let's move on. Okay, so I'm just gonna teach you guys a little short segment, okay, of nucleotides. So a nucleotide is nothing but the simplest form of our genetic material. What comes into your head when you think of genetic material? DNA and RNA, right? So there are polynucleotide. DNA and RNA are polynucleotides. So what's the monomer of these polynucleotides? A nucleotide, yes. So we'll learn so the definition of nucleotide is very important. Subunits that make up DNA, RNA are known as nucleotides. Subunits make up DNA and RNA are called as nucleotides. Therefore, DNA and RNA are called polynucleotides. Right. So this is how a nucleotide looks like. Easy. This is a ribose or deoxyribose sugar. This is a ribose sugar in case of RNA. In case of RNA. And it's deoxyribose sugar in case of DNA. In case of DNA, my bad. Yeah. Okay. The nitrogenous base is same for both DNA and RNA. So based on the nitrogenous base, it's gonna be either adenine, guanine, thymine, or uracil in case of RNA, or ATCG, cytosine. So I'm just gonna write it down here. ATCG or AUCG in case of RNA. In case of DNA, And phosphate group, non-negotiable, it stays. Okay, let's move on. Now, we already learned this in the diagram. So nucleotides are composed of a nitrogen containing base, also known as the nitrogenous base, and then a pentose sugar. Very important, pentose sugar. So a pentose sugar could be ribose or deoxyribose, and then you got a phosphate group. Alright, guys. ATP. At this level, most of you guys are aware of what ATP is, the energy molecule, and ATP is actually a form of nucleotide. All right, and it is formed by a phosphorylation process. Basically, AMP is adenosine monophosphate turns into ATP, adenosine diphosphate turns into ATP that we know, adenosine triphosphate. So knowing the full form is as important as knowing what it is. So you have an ATP, right? You have an ATP molecule. What happens if I remove a phosphate from here? A single phosphate. Triphosphate means what? It has 3 phosphates. What happens if I remove one phosphate? It forms ADP. What happens I take this ATP, adenosine diphosphate means di means 2 phosphate. If I take another phosphate away from it, it turns into AMP. AMP, adenosine monophosphate plus phosphate. Okay, so this whole process this process that happens from here to here by adding phosphate is called the phosphorylated nucleotide. Basically, the nucleotide is formed whenever we add a phosphate. So let's learn a little bit. Let's recap. ATP, energy carrying molecule that provides energy. It's another type of nucleotide, hence it is structurally very similar to the nucleotides that make up DNA and RNA. Let's go move on. Alright, for our purine and pyrimidine bases. Purine, pyrimidine bases. The purine bases are the bases that have double ring structure. 2 rings, and pyrimidine bases are bases that have a single ring. There's only 2. We have 4 nucleotides. 2 are purines and 2 are pyrimidines. What are pyrimidines? They have double ring structure. Purines and pyrimidines, they have a single ring structure. I'm going to recap. Purines, double ring. Pyrimidines, single ring. So purines, example is adenine and guanine. And the pyrimidines are cytosine, thymine, or uracil in case of RNA. So uracil is the replacement of thymine in RNA. So in DNA it's thymine, and in RNA it's uracil. So in DNA it's ATCG, and in RNA it's AUCG. Alright. For your exam, both at Excel and Cambridge, knowing these diagrams are very, 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 very important. And unfortunately, I can simplify the other topics, but this is just one thing I can't simplify. You have to you really need to know how to draw these. How to draw these, exactly how it is, especially knowing how many nitrogens there are. For example, in guanine, we have 1, 2, 3, 4, 5. We have 5 nitrogens. But in adenine, we got 1, 2, 3, 4, 5 as well. But main difference is, for example, in guanine we have a C double bond O carboxyl group. But in adenine we don't have that. Adenine and guanine have similar two pentose sugars, as well as guanine has 2 pentose sugars. Adenine has 5 nitrogens. Guanine has 5 nitrogens, but guanine, the only thing that differentiates guanine is this group. I will just highlight it in a different color. This group, C double bond O. Okay, this is for adenine and guanine. Okay, that's how you will know. When you're drawing, you will know you're right. Thymine and cytosine and uracil have single pentose sugar. Thymine, as the diagram is a bit tough. As you can see, there is 1 nitrogen another nitrogen. There's two nitrogens and there is two carboxyl groups. This is how I usually try to remember. So thymines have two carboxyl groups and two nitrogens. And then it has a methyl group. Those of you guys who are studying chemistry will be very familiar with this. This is a methyl group. Alright. Cytosine is a bit much more simpler. It has, similar to thymine, it has two nitrogens. But oh wait, not similar to thymine. Thymine has two nitrogens and cytosine has three. See, notice three. But it has one carboxyl. So this is the technique of remembering. Each cytosine has how many nitrogens, how many hydrogens, how many carboxyls. This is how you're supposed to remember it. That's the easiest I can think of, honestly. The same thing goes to yourself. How many nitrogens? You got one and you got two. And then you got two carboxyls. Alright, students? Very, 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 very important. Practice this. Really, really hard for your exams. Structure of DNA. I have nothing, honestly, it's pretty much familiar to you guys. Adenine always binds with thymine. Cytosine always binds with guanine. So when I was a student, I used to remember it as ATCG. Adenine with thymine, cytosine with guanine. Watch something. The hydrogen bonds. This is a very important exam. Adenine and thymine forms how many hydrogen bonds, and cytosine and guanine forms how many hydrogen bonds. So adenine forms two hydrogen bonds with thymine. Cytosine and guanine forms three hydrogen bonds. So adenine goes in Ta. It's just flipped. Ta, thymine and adenine forms two hydrogen bonds, and guanine and cytosine forms three hydrogen bonds. So if I write it down, so adenine and thymine forms two hydrogen bonds, and cytosine and guanine forms three hydrogen bonds. Our students, what I already explained is already in here. So purines, adenine always pairs with pyrimidine thymine, forms two hydrogen bonds. Guanine always pairs with pyrimidine cytosine, forms three hydrogen bonds. This is known as complementary base pairing, and they form DNA base pairs. So this is just a table that differentiates between DNA and RNA. Usually they will say compare the DNA and RNA. So DNA is deoxyribose, RNA is ribose. We'll learn how will we understand deoxyribose and ribose. As you can see here, ribose has this extra OH and deoxyribose doesn't has that OH. Other than that, the whole structure is same. The whole structure is same. Only difference is they have on carbon 2 position, they have an H, and ribose has an OH. Basically, this is I don't know if you guys can look at it, but this is supposed to be O4 and this is supposed to be O5. So they have one less oxygen. That's why we call them the DNA deoxyribose, and the RNA ribose. The DNA, I already told you guys, ATCG, so adenine, thymine, cytosine, guanine, and they have adenine, uracil, star, and then cytosine and guanine. DNA is double-stranded, basically double helix, very important. What's the structure of DNA? It's a double helix. It's an RNA single-stranded. Alright guys, that was a pretty long class, wasn't it? Thank you guys, whoever you guys are in the live session. I can't really see, but I hope you guys enjoyed this live session. Thank you so much for paying attention. Hope you guys study hard, work hard, and